It's really nice to be here, and I wanted to start exactly at the point of talking a little bit about my past and then where I am now. So thanks for the introduction, Megan. So these are, this is where I started this journey that we should talk a little bit about. And by the way, please interject if you have questions. We're a small group, so we can make this informal. So I started at Notre Dame, and I really had a passion for science. I was a biochemistry major when I was here, and I did a theology minor. And then I went to Chicago for medical school, as Megan said, and this is where I still live and do my work and so forth. But there was many sort of paths in between, and I thought that since you all were getting ready to embark on the same journey, it might help you to see what sort of my path has been. There's no one path to pursuing a career in science. I purposely titled this slide that way because that's really how I looked at it. I was passionate about science, I loved doing it, and so I wanted to find a way to have a career where I could get to do that every day. And there's lots of different ways to shape that. I mean, how many of you are thinking about medical school? Okay. How about grad school? Okay. How about sitting on your parents' couch? Right. So some people might not have it figured out yet, or doing some other work for a couple of years before they figure that out. But what I think it's about is find something that you want to do for a living that you actually like doing, right? And then figure out if somebody will pay you to do that. That's right. I mean, it makes your day-to-day life so much happier and better if you really can do something that you love doing, because then it doesn't seem like work. And I've managed to find that, and I'm really happy that I did. So my path was this. So at Notre Dame, 98, and then I actually took a little bit non-traditional medical school path. So I did go straight to medical school. A lot of people, will do things before they go to medical school to gain more research experience. I went straight to medical school, but then I did 5 years in med school, as opposed to the usual 4. I didn't get a PhD, but I took a whole year for research fellowship. So I put it on here. It was through a Howard Hughes fellowship for a year in between my between my second and third year of medical school. And during that time, I actually went out to Los Angeles, California, because there was a lab out there that I was doing sort of the exact same kind of science that I wanted to study. So I really wanted to submerge myself in a lab for an entire year to learn that side of doing science. I do it every day, for a year. And it's interesting how things can always connect, right? So when I was here, it was a Howard Hughes summer fellowship, right? It allowed me to do my first real summer of research. And there's only one summer you get in medical school. I don't know if you guys know that. Otherwise, the years are year-round. So after the first year of medical school, you get the summer to do whatever. And of course, they expect you to do something, to continue your learning, to do some kind of research. So I actually got a Howard Hughes summer internship during that year of medical school, and I did that in this lab out in California. So Pritzker was, really open-minded. They said, as long as you're doing something that you want to do, go for it, if you can find a way to make it happen. So I found this lab and went out there for the summer through this Howard Hughes program and liked it so much. It's crazy how things happen, and I never really planned it. But one night I was in the lab, it was like 8 o'clock at night, and my PI comes in, right? And he says, I think he just sort of saw me there, and he's like, why is this summer student here at 8 o'clock at night, right? Why does he care so much? And so he said, what do you want to do? And so we sort of talked about it a little, and I told him how much I really wanted to have a career doing science, right? And he said, why don't you take a year off of medical school? I had never thought about that. It sounded crazy, right? Why? Medical school is already long enough, right? And so the more I thought about it, I said, I really could gain a lot from doing this. So I applied and got a Howard Hughes 1-year fellowship, right? You have to find ways to sort of fund, being able to do this. And so I was lucky enough to get that, and that's why I did the 5 years. So I said that to say that, look how this sort of kept coming up. I had Howard Hughes summer research, a second one, and then it led to a year-long. And I guess my point is, you guys are at a great school, right? And the hallmark of grade school, I don't think, is always just about what they're teaching you, but the opportunities they give you, right? You get opportunities that other students don't. You're in an honors program, you're getting opportunities that the regular students here even don't. So take those, take those opportunities, pursue it, not to build your resume, but just to say, I want some experience in this, because if I'm actually going to do this for a living, I got to find out if this is something I really like doing. So if I can leave you with anything today, it's that, and you never know where it might lead. Okay? So that was sort of my journey, and it really set up everything that I ended up doing. I can't tell you how many times people ask me about that year that I took off in medical school and what I did during that year. You know, every other time you then start doing interviews for all of your other, you know, trainings, people they want to know. They want you to.
It's not very common, right?
Correct. Correct. Not that common, right? So that catches people's eye, right? And they say, well, what did you do during that year? And then it leads to a conversation. And if you can talk intelligently about the research you did, and they say, hey, this person's pretty interesting, right? So I never once did it to build a resume. I never thought about it that way. It was more about, I liked it, and I wanted to see if I really wanted to do this for a living. And then people noticed, right? They say, well, he took this opportunity, and he did this. And so that sort of is something that distinguishes you, right? So then I went off to, after medical school, I went off to do my internship in internal medicine. That's 1 year, you probably know. And then the residency is 2 additional years. I actually did my internship at Chapel Hill in North Carolina. I had been in Chicago for, you know, medical school, and I felt like I needed to get a different experience, right? I wanted to see if hospitals work differently somewhere else, right? So I went to Chapel Hill. And then my wife, she was behind me in the training, and she also was matching in a career as a physician, and so we ended up then coming back to Chicago where I finished my residency. And then here's, you know, one more chance to sort of pursue a path or take an opportunity that might get presented to you. So I always knew I wanted to go into oncology, so I went to a fellowship in oncology, which normally is 3 years, and I actually decided to do 2 fellowships, another fellowship concurrently in clinical pharmacology and pharmacogenomics, because I really wanted to learn about drugs. So I remember it hitting me one day when I was in first year of medical school. I said, well, what kind of doctor do I want to be? What do I want to study for a living, right? And I was actually in my pharmacology class, right, in first year of med school. And I said, you know what, I'm really fascinated by how drugs work in the body. Right? So again, it comes back to find something that you're really just interested about. And so I always knew I wanted to see how drugs worked in oncology, and I also wanted to figure out if I could, you know, get some extra training in that to really be able to do it for a living. So that's why I opted for a second fellowship, and I was lucky because you could add this up. I mean, it could have been 5 years of fellowship, but they let me do it in 4 years. They gave me sort of one of the years as a concurrent year. So if you add it up, I mean, it was 12 years more of training after Notre Dame, right? It sounds like a long time, but each step was, you know, it's something different, and I was gaining some skills to let me do, you know, what actually I ended up doing every day. And that is, you know, so I'm a physician. I treat patients with cancer. I spend about 20% of my time doing that. I love taking care of patients. It's the reason I became a doctor, right? I love taking care of people, helping people, you know, one-on-one, seeing a person get better when they have cancer. It's a tremendous feeling, right? And so then the rest of my time, so you're saying, well, why? That's only 20% of what is time. So I actually get to teach. So I lead the pharmacology course that I was telling you about, right? I direct that course in our medical school now, and then I do research, and that's what I'm going to spend the rest of the time, you know, talking about. But really, this whole career of being involved in research during different steps allowed me to say, I want to have a career where I'm doing research for most of it. So that's what I'll talk about. All right. Okay, so the rest of the time we'll talk about my research, and you know, I put that part in there about a career path so you guys could think about different ways to do this, right? There's no one path, but different ways you can piece parts of it together. All right, so before I talked about my research, you probably hear people who often have to disclose any potential conflicts that they might have, right? So I've now developed some conflicts. I don't know if this is a good or a bad. So one is that the technology that I'm going to tell you about that we ended up developing, the university, so when you work at a university, anything you invent is the university's property, really, right? It's not your own property. So the university owns it. It's their intellectual property. So the university decided to file a patent for this genomic prescribing system that I'm going to talk about, so I'm named as a co-inventor on that patent. And then, I mean, within the past month, the university has decided that they thought this technology was so cool that they wanted to try to commercialize it, right, and form a company. And because, you know, there's more than one way to try to, you know, affect the world, right? One is you can do that by publishing papers and trying to convince people that your ideas are learning something new about the world. And there's other ways to try to sell something that maybe to hospitals or doctors that would help them take better care of their patients. So the university has founded this company called GojiMed. I don't even think we have a website yet. If you look at it. This was literally within the last couple of weeks, and so I'm considered a co-founder of this company that's going to sort of do the same thing I'm talking about. Okay. All right. So what is personalized medicine, right? My talk was titled Personalized Medicine. What does that mean? So I was really struck by this, right? When the diversity of people, right, that I would see in the clinic. And when I would take care of patients, I would notice patients with the same disease, I would give them the same drug, and two widely different things would happen, right? One patient would do really well, they would respond to the drug, their cancer would get better, another patient wouldn't work. One patient would have severe side effects, another patient none at all, right? How can that be? So I was really struck by that question. Again, I'm coming back to that, find a question that you're passionate about and then pursue it. So I was struck by that question, I said, this is what I want to figure out. And I thought, in our era, where the genomic era is booming, right? There's all this information about what makes us different genetically from each other. I said, there's got to be an explanation there, genetically, that can explain what I'm seeing, why two people do so differently. So it really came from an observation that I felt in the clinic. And you've probably heard about this term called personalized medicine, or precision medicine. Right, our president's talking about this. Is from the State of the Union address just in January, where he had launched this precision medicine initiative to basically try to do that, to try to give access to personalized information to keep ourselves and our families healthier, to try to really understand what makes us each different, why we might respond to drugs differently. Okay, so that was, it's a timely issue. All right, so pharmacogenomics, all right? This is the sort of subject of what I'm going to talk about. There's a definition up here. Anybody ever heard this term, pharmacogenomics? Some people have, I've learned. If you see me abbreviate it later in the talk, I abbreviate it PGX. So it's really this study of how genetic factors can explain differences in drug response or drug side effects, all right? And this, I would argue, is only one piece of personalized medicine. What do I mean by that? So what personalized medicine? Treating a patient personalized. Anybody suggest other factors?
Could be like their specific behaviors of the environment.
Yeah, right? So can you give an example?
Like certain dietary things. So if you eat a certain amount of vitamins, that could affect how a drug would interact with the cells in your body.
Very good. What's your name?
Sarah.
Sarah, excellent. So environmental factors, right, that make us different. All right, how about other factors? Go into a personalized medicine decision.
I think just interacting with the patients themselves, knowing their routines, their family unit, I guess. Just what kind of support they have as well.
Great, Megan. I mean, you'd be surprised how much of a difference that can really make in how well a person does or what side effects they might actually have, is because some patients will notice something and they have someone there to call the doctor and say something's going on, and other people, no one calls, right? And it becomes a severe problem. So that's a big factor. How far a patient lives from the hospital, it's incredible how much of a difference that can make, right? And even in deciding what kind of drug you might use, because if you need a drug that requires someone to be followed very closely and they live 3 hours away, that's not very practical, right? So those factors are important. How about other things?
Medical history.
All right, what do you mean by that?
Like past conditions that they've had, drugs that they're on or have been on, and how they reacted to those, and other factors that might influence what drugs you prescribe or something.
Great. What's your name?
Gary.
Gary. Great. So you said a couple things there in my view. One is other comorbidities, other medical problems that they have might impact the choice of a drug. Right, a drug that you're thinking of for their cancer might be really a bad idea for their heart disease. Right. So you got to know the other factors that the patient already has in their medical history. And then you mentioned other drugs, right? So what are you getting at there?
Like drug interactions,
Yeah.
or if they had a bad reaction to something similar before, to
Great.
not prescribe that type of thing.
Great. So that's an important distinction, right? So drug-drug interactions are an important piece. I'm going to talk about drug-gene interactions, right? But you can't ignore the drug-drug interaction. It's a great one. Any other factors? Yeah.
Socioeconomic status and like the cost of whether they have insurance or not.
Okay. Yeah, it does come into play. You're right. Thankfully, not that much,
Yeah.
surprisingly. For me, in practice, usually, the drugs we're prescribing, the insurance pays for them. If they're making a difference to a cancer, insurance companies usually don't want to deny that, right? So they find a way to be paid. But that's important. Yeah, sure.
And you can also consider how likely that person is to take a drug. So kids and antibiotics, they tend to do the Z-Pack because it's a shorter duration and you're less likely to forget, whereas someone who is more responsible or follows a bigger routine could maybe take something, be relied on to take something more regularly than someone who doesn't have as strong of a routine.
Mm-hmm. Very good. So yeah, the adherence, right, that a patient might have. Very good. That matters. Anything else? Yeah.
Family history.
Oh, good. Yeah. What do you think family history is really telling you, though? It's a good one.
It's not as like a direct measure of like genetics if you're going to look at this. So this would be more direct to see if you had a predisposition to a heart disease, but you would kind of have that abstract look at it if like a parent of the patient had something like that.
Exactly right. What's your name?
Josiah.
Josiah. So great point. You know, I think of family history as a proxy of genetic history, right? That's really what it's trying to do. If we can't get genetic information on everybody, we're trying to get at that, right, through our family history. So that's really good. All right, good. Anything else? Just, I mean, you guys have said so many good ones. Anything just really simple about, you know, you looking at the patient in front of you and something you might think about to make a personalized decision? You guys have said almost all the things that I could think of. You were getting at it a little bit with Sarah, with maybe kids versus adults. What am I thinking of?
Well, like, the way you would tell, I mean, some people are more able to handle scarier news.
Okay. All right.
Necessarily, so sort of that bedside manner aspect of how you tell someone, here's what's wrong, here's how we're going to treat it, and like giving them enough information so they understand it, but not overwhelming them. Okay,
good. So bedside manner, the way you explain things. How about the difference between a child and an adult, just physically? Okay, so does body size matter? Right? Does gender matter? What about, you know, organ function, right? What if their kidneys don't work very well and this drug requires kidney elimination, right? Or they have liver problems, right? Gary, you were getting into a little bit of that. So you've got to know all these factors when you're going to prescribe a drug, okay? So we spent 5 or 10 minutes on this topic, right? So the point of that is when you're talking about personalized medicine, it's not just genetics, right? It's all these factors we're talking about, and genetics is one extra piece of that maybe we can now add to the equation, but you can't ignore all the other factors that go into personalized medicine, everything you've said. So if you hear people say that, you know, personalized medicine is something new, I'd say physicians have been practicing personalized medicine for centuries, right? Just in a different way, right? We've now acquired maybe another tool.
Can I ask a quick question, just because of the talk we heard this morning, who was a PhD geneticist? Okay. What, if you had, and maybe this is an unfair question, what % of a information is the genetics versus everything else?
Yeah.
How important is it, or how unimportant is it?
Yeah, great question. So for drugs, we'll talk about drug prescribing. So for drug prescribing, I can tell you that the best example we have is a drug called warfarin. It's an anticoagulant, right? Where they figured out that basically 2 genes explain about 50% of the dosing variability, okay? And the other 50% they haven't been able to figure out yet. It's probably all these other factors that we're talking about. So that's you know, probably one of the drugs where, you know, the genetic factor is one of the biggest. I'd say for other drugs it's probably even smaller than that, right? The genetic factor. It's only one piece of it. That's a good question. All right, so what are the potential advantages of using pharmacogenomics in practice? All right, so first off, people know what the Hippocratic Oath is? What is it? Do no harm, right? So we all take this oath. First, do no harm. Turns out we're doing harm in the drugs we give, right? So the statistic is in-hospital death, patients dying in the hospital. 5th leading cause is a drug adverse reactions that we've done. It's terrible. What about the flip side? Do the drugs we give work for patients? It turns out only about half the time. So we look at the most common diseases and the most common drugs to treat them, and we do, you know, large analyses of patients and telling us whether these drugs have helped or not. Only about half the time do patients say that the drugs actually appropriately treat their condition. So we're not doing very well on either side of the coin. And then third, you could look at this economic benefit of pharmacogenomics, right? We're wasting, you know, so much in our trial and error approach, right? Well, we'll try this drug, and if it doesn't work, we'll try a second drug. And if it doesn't work, we'll try another one. You've wasted not only time to the patient, but the cost of all those drugs you prescribed. And then what about if you prescribe a drug that has an adverse reaction? What does the patient do usually? They might call you, but what else might they do?
Stop taking it.
They might stop taking it. That's right. But I think about a downstream cost. Where would they go if you have a problem in the middle of the night? You just took this pill. Yeah, they go to the emergency room, right, where a bunch of tests are then run, driving up the downstream costs of healthcare, maybe for an adverse reaction that could have been prevented or identified in advance, right? So people use these as arguments for why pharmacogenomics. All right, so I like to put it into a context of a couple examples. So let's talk through a couple examples. So people heard of this drug, Simvastatin? Okay, it's the number 2 most prescribed drug in the US, 83 million prescriptions a year. I would venture to guess that in this room, multiple of you have parents that are on this drug. Okay? It's one of several drugs that treat high cholesterol, hypercholesterolemia, and other alternative drugs include other drugs in the same family, other statins, okay, and then non-statin drugs. The major limitation of Simvastatin is myopathy. Anybody know what myopathy is? You figure it out from the word? Yeah, very good. Josiah, did you say that? Yeah, what was your name?
Dennis.
I couldn't tell. What is it?
Dennis.
Dennis, very good. So it's muscle pain, right? So people who get myopathy, they'll just complain of this generalized aches in their muscles all over, and it's usually bad enough where they stop the drug. They figure out pretty quickly, it's this drug causing it, and then they stop taking it, right? And then you don't control their cholesterol. In the most severe forms, if you get myopathy, the muscle breakdown, so the actual intracellular release of contents of the muscle can cause kidney damage in the most severe form. So myopathy is something that we want to avoid. All right, so is there a genetic predisposition to developing myopathy from Simvastatin? It turns out there is. So this was the landmark study. So New England Journal of Medicine is probably the most preeminent journal in all of medicine, right? So the biggest, most impactful studies are published in this journal. So you can see this was from 2008. And the title of the article was SLCO1B1, which is a gene, SLCO1B1 variants and statin-induced myopathy, a genome-wide study. Okay, so let's go through this. So I'm a big believer that if you're going to have a career in science, you've got to learn how to read graphs. Okay? You've got to learn how to interpret data. So let's see if we can go through this one together. So the y-axis is cumulative percentage of patients who had myopathy, and the x-axis is years since starting Simvastatin. And there's 3 curves on the graph, right? 3 different genotype groups. Okay? One genotype group where people have the CC, one for the heterozygotes, and one for the homozygote recessive, right? TT. Actually, do we know that that's the recessive? I actually misspoke. We can't tell, can we, how many people are in each group here. So the homozygote group with that's lowest on this curve. All right, so let's see who wants to take a stab at it. What do you interpret from this? Could be more than one, interpretation. Okay.
So, excuse me. After taking the drug, the TT genotype basically has the lowest percentage of myopathy.
Okay.
Whereas the CC genotype has the highest, and then the heterozygotes is in the middle.
Okay, very good. What's your name?
Lupin?
Lupin. Okay. So you gave a perfect explanation of that. Would you say that this suggests that there's a pharmacogenetic relationship here? Does this genotype seem to impact the outcome with this drug?
Yeah, I say so.
Okay, yeah, it seems like it, right?
Yeah.
The data seem pretty convincing. All right, good. Let's dive a little deeper. What else does the graph show? Go ahead.
It's a staggered effect. So as the years go on, the number of people who get myopathy increases, like pretty, I guess, logarithmically almost. I don't know.
Okay, so it does seem like that the longer you're on the drug, the higher your risk. Can I rephrase it that way? Okay, that sounds good. Although, if you're going to get it, if you're going to get myopathy in any one of the groups, it seems like, most of the people who end up getting it, it happens pretty early, right? I mean, even if we look at the high-risk group, it's within that first year. There's not that many extra people where it's happening out in later years, right? Is that fair?
Yeah.
Okay. Yes.
It's like not a proportional change. It's like the presence of one just causes may still an increase, but not as much as the presence of both of them.
Great. What's your name?
Zoe.
Zoe, great observation. So I think this is really key, that there seems to be, I call it a gene dose effect here, right? Doesn't it seem like there's a gene dose effect? And I don't mean the dose of the drug. I mean the number of copies of the gene, the allele you have here, seems to matter what your risk is, right? And one allele raises your risk a little bit, right? One copy of the C allele, but 2 alleles really increases your risk, right? Excellent point. All right, one more. Yes?
Just because you have the CC genotype doesn't mean you'll get myopathy.
Why? This is the hardest one to notice, and you noticed it. How do you know? You're right.
The y-axis one goes up to 20%.
Right. Look at your y-axis. Look at the numbers, right? So even people in this highest risk group, CC carriers, only 20% of them are going to get myopathy. Right? So what does that mean? Is there still a genetic relationship here?
Compared to the other two, I would say yes.
Yes.
But it's not like 100% guaranteed.
Okay, so what other factors are present here?
Anything we've talked about.
Everything else we talked about, right? Exactly. So this is the point, right? That genetics are only probabilistic, right? It doesn't mean you're going to exhibit the outcome if you carry the genetics, right? And you've always got gene plus environment, right? The classic question of how much genetic influence, how much environmental or other factors, right? What this study can't tell us from this graph is, well, do we know anything else about other drugs that might impact myopathy risk, kidney function, which might impact myopathy risk, age, which might impact myopathy risk? Turns out all three of those matter, okay? They don't tell you that on this graph, but those are other factors that you would want to know about. All right, so how many people, since seeing this, would say, all right, pretend you're the doctor. Would you want to know this information about your patient before you prescribe Simvastatin? You see people shaking their head. Anybody see any downside to having this information? Yeah?
If you knew about it and you accidentally prescribed it anyway, you could be liable.
Okay, yeah, liability risk, right? If you knew about the genetics, great point. We'll get into that in a little bit.
There's still a chance that even if they are like TT, there is a very small chance that they'll get myopathy anyway.
Okay.
So even though you like took proactive measures and prescribed this.
Okay, so maybe
still,
like a false reassurance or
Yeah.
something like that. Yeah, that's a good point. Good point. So
Are there other good like similar like effectiveness for like treating this high cholesterol, or is like this the main drug? Because then like it would be kind of hard to be like, well, there's a 20% chance of them getting this, but like the other drug is like so much less effective.
such a relevant question, right? Really great, Zoe. You know, what are my alternatives? If I'm not going to prescribe Simvastatin, do I have another option? You got to ask that, right? That's really great. And what about maybe the other related question? All right, say I don't prescribe this. What if I say, all right, my patient's in this high risk group. I don't want to give them Simvastatin. I don't have another alternative. Well, what's
when they have high cholesterol and have complications from that.
Yeah, so they have a heart attack because their cholesterol is too high or something, right? So you have to think about the implications of not treating, too. So this is, see how complicated it gets, right? You can't just use genetics in a vacuum, in isolation. These are all the other factors why you go to medical school for 4 years, right? To just consider all these other factors about the patient sitting in front of you. And your decision might be different for Megan versus Sarah versus someone else, right? That's what goes into the personalized medicine decision. Want to do one more example? All right, so let's take clopidogrel. So clopidogrel, anybody ever heard of this? Plavix is the brand name. Okay, you maybe see commercials or something. So clopidogrel is actually a prodrug. It's taken as a pill, gets absorbed in the intestine, and quickly, majority of the drug actually gets broken down by esterases into inactive metabolites. So 85% of the ingested dose gets quickly inactivated. Okay, so only 15% is left available for actually doing the job. And so the part that then it's absorbed in the blood, the part that then gets metabolized in the liver through hepatic metabolism actually gets activated. So the remaining 15% gets activated. And when I say it's a prodrug, by definition a prodrug means that the form you're taking in is not the active form, right? So it has to get activated in order to do its job. And so clopidogrel is an antiplatelet drug, okay? So it works on atherosclerotic plaques in arteries and tries to prevent clotting, right? So you can imagine that if you have a clot that forms in here, that's what causes a patient having a heart attack. So it turns out that you've got to have certain genes, certain enzymes present in your liver to properly activate clopidogrel. And the most important one is this one called CYP2C19. You guys heard of the cytochrome P450 genes, enzymes? Okay, so this is one of those. It's a common one. All right, so you need that to be fully functional to activate clopidogrel. So, could there be a genetic inherited risk to not being able to activate clopidogrel? Well, here's the study. So again, published in the New England Journal. This was from 2009. Okay, and so I'll sort of set it up and then let's talk through what it means. So the y-axis here is the primary efficacy outcome. They were measuring the efficacy outcome. What was the primary efficacy outcome? It was either death, heart attack, or stroke. That sounds like pretty important outcomes, right? Being measured. And then the x-axis was days since randomization. And you can ignore the randomization part. This was part of a larger study where they actually randomized patients. But everybody in this graph that they're showing took clopidogrel. All right? So there's two curves here. One is a curve called carriers and another group called non-carriers. And I'm not going to tell you what it means yet, but let's talk generally about what people see here. Who wants to tell me what they think the results are showing. This is a harder one.
If someone who's a carrier has more chance of having death, stroke, or heart attack.
Okay,
But
good.
it says 53%, but it's just like, it's not actually like a 50%. It's a 50% of the non-carrier gene. It's not
good.
50% of like nothing.
Good, Sarah. Very good. So you hit on a couple of things there. One is that, all right, this seems to show that the carriers, the people in the red curve, have a higher risk of death, heart attack, or stroke on clopidogrel. Remember, everybody takes clopidogrel in this study than a non-carrier. And you pointed out that, yes, it's a 53% higher risk compared to the non-carrier group. You're getting at a relative risk there. People call it a relative risk relative to something else. What's the absolute risk? They tell you here.
Well, for carriers, it's 12.1, and then non-carriers is 8.
Yes, exactly. So if the patient's sitting in front of you and they actually ask you, well, what's my risk, doc, of having a heart attack or stroke here? You could tell them, right? It's like 12% or 8%, right? So that's the absolute risk if you've heard of that term, okay? They both numbers matter, right? All right. So what do you think's going on here? What carrier, non-carrier? So this is a pharmacogenetic study, I'll tell you. How can we relate it to CYP2C19? Any ideas? Yeah?
The carriers have one copy of a gene that is somehow affecting the outcomes of this, whereas the non-carriers are probably homozygous for the normal, I guess, of whatever that gene is.
You got it, Gary. How did you figure that out? So, based on the previous slide I told you?
I guess kind of.
Yeah.
Just that carrier is generally a heterozygote.
Yeah.
And often used in the connotation of something negative.
Good, yeah, good. So going back to some of your genetic roots here, what you've learned, right? So it turns out that these people in the red group indeed carry a polymorphism in their inherited DNA that inactivates the enzyme, one copy of the enzyme, right? And so if you don't have a fully functional enzyme, you can't convert clopidogrel, right? If you can't convert it, it's not in the active form, right? So those people then would be at a higher risk, right? Because clopidogrel is not activated like it is in the group who are wild type, the non-carriers who have normally functioning enzymes. You got it exactly right. So now it makes sense. So is this a pharmacogenetic relationship? Does it look like there's a pharmacogenetic effect on this drug? Yeah, I mean, there's clearly a difference in the two curves. It was statistically significant. Is it clinically important? What do you think? Yeah, Josiah?
I think definitely because if the actual effect of this drug is dependent on the function of the enzyme, I forget what it was, but I was on something and there were two different types of the drug, and I messed up telling someone what the prescription was. So one was the already metabolized version of it, so the active form, and then the other one was just what would be metabolized. So we were talking about it for clinical significance. The one that was the metabolic product that was active was hundreds of dollars more expensive even with insurance. So.
Okay. Yeah, that's very good. Was it about this drug? Are you talking about?
No, it might have been. I can't remember.
Okay. All right. That's very good. All right. So how many people? You have a question?
Yeah. I was just wondering what the curve was for someone who didn't take.
Yes. Great. I'm glad somebody asked that. Right. So what if you don't take any antiplatelet drug at all? Right. It turns out their risk is about 20%. So the drug still works in people who only have partially functioning enzyme. Right. Yes.
Has there been any study on, not the carrier, but both genes being?
Yes.
affected?
Yes, so this study actually included patients like that. They're rare, those kind of patients are rare in human existence, right? But those people are actually included in this red curve. So they put people in this red curve who either had one or two non-functional genes, right? Or reduced function genes. So good question. All right, so how many people would want to have this information, you're the doc again, about their patient before they prescribe clopidogrel? So seems like a no-brainer, right? I mean, why wouldn't you want to have this information? So what would be the questions you'd want to ask? So Zoe was getting at this a little bit. So say you want to have this information, what else would you need to know to make the decision about prescribing this? So what's the risk if you didn't give clopidogrel at all? And then you were starting to get at this, I think, Josiah, about alternatives, right? So what do people think there?
Yeah, the cost of this drug versus the cost of alternatives.
Okay, good. So an economic argument. Pretend we're practicing, a hospital system where they don't care about that. They say do the right thing for the patient. So what is the other question you need to ask? Well, is there an alternative? Right. That's first. What else might you ask? I'll tell you there are two alternatives to using this that are equally effective to clopidogrel in all comers. What else might you want to know about those two drugs?
Are they going to be better for people who are carriers?
Yes. Right? So now we have an option for the carriers where we could give them the alternative drug and we know it's going to be effective all the time. There's one final question that I'd want to know before I implemented this type of testing, right? So say I'm a cardiologist and I know about this, I read this paper, I want to use it in my patients. You were getting at it a little bit, Dennis, when you asked about people that have two non-functional. So if we're going to put into a widespread implementation of maybe testing everybody before we prescribe these drugs, what else would we want to know about the population regarding genetics here?
How common is it in general?
Yeah, how common? Can you tell? This graph actually tells you a little bit. Look at the numbers.
Most people tend to be a non-carrier, looks like.
Okay, so most people are non-carriers. That sort of makes sense, right? I mean, would this drug have ever made it onto the market if most people couldn't activate the drug, right? It probably wouldn't have been a very good drug. So then it makes sense that most people are the non-carriers. But can you see the numbers? It looks like 1064 in the non-carrier group and 395 in the carrier group. So can anybody do the math quickly? What percent of the population has at least one copy of a non-functional or reduced function allele? Roughly.
30-ish%?
Yeah, 25, 30% of the population, right? What do people think? Does that sound like a pretty important size number? Yeah, I mean, look at this room here, right? 2 or 3 of us in this room are in the red curve. Okay? Would you want to know before you got this drug? Right, probably. All right, so you get the idea. Yes?
So what's the argument for using clopidogrel at all if the other two drugs don't?
Are they as effective?
Great question. So yeah, so it turns out the other two drugs are as effective as clopidogrel, but they have a higher bleeding risk. So using them in everybody, you increase your bleeding risk. They also happen to be 6 times as expensive. All right, so let's talk. It's already 2:30 here, so we got to move forward into actions. I can show you some of the research we've done. So what fascinated me was saying, all right, all these kind of findings have been published. I've shown you 2 examples. There's dozens of others more like this about pharmacogenomics. Has anyone ever gone to a doctor where they've ever brought this up? No. And I can tell you, I speak to audiences of hundreds and thousands of physicians, and I ask for a show of hands of doctors who ever ordered a pharmacogenomic test. 1 or 2 hands will go up in the room, right? So it's not something that's commonplace, and I'm struck by that, and sort of said, I want to try to change that, right? I think we're going to look back at this, era of medicine, saying we need to consider this kind of information, individual information, before we made a prescription choice. And we're going to look at it as being so archaic a way to practice medicine. So I think that the time has come to interject genetics into this clinical decision calculus, right? Because I can tell you, docs feel like this. They're at this point where they've got usually multiple options to treat the same condition, and they scratch their heads saying, oh, I'll sort of pick this one because it's one I'm familiar with, but you have no idea knowing whether it's the right choice for you. Okay? So that's where we talk about precision medicine actually being implemented. Are we ready for this? And this was a survey a few years ago, over 10,000 US physicians. 98% of docs said they believe genetic profiles influence drug therapy. So if you're convinced by the 2 examples I've shown you, you're not alone. Most docs say they realize this is important. What are the real barriers to doing this? Can we spend a minute thinking about this? So we mentioned one just now, that docs maybe are ill-prepared for integrating pharmacogenomics at the bedside. Why else is nobody ordering this yet?
Maybe, I don't know how long it takes the time between you test it and getting it, if it's like require immediate treatment.
Great, right? The patient's sitting in front of you, they want a prescription for their condition, right? They don't want to wait days or weeks for you to say, well, I need to order a pharmacogenomic test and I'll get back to you, right? It's not practical. So we'll show you how we try to get around that. Zoe?
Isn't it also kind of like the ethical considerations where it's like, I don't know, like maybe you don't want to know if you, like, no drug is going to work on you. And also at the same time, do you want your insurance company to know stuff like that as well?
Great point. So somebody usually mentions this point when I talk about this. So let me try to address it a little bit. So one is that I think there's two kinds of genetic testing, right? Pharmacogenetic testing might be a little different than other kinds of genetic testing because it's only telling your risk of what happens to you with a drug. And the patient still has the control of whether they're going to take that drug or not. And almost always there is an alternative, I can tell you. Okay, so those concerns should be less for pharmacogenetics than they are for disease risk genetics. I mean, there's a lot of people that feel that way about, well, I don't want to know if I'm going to get Alzheimer's or breast cancer, right? Okay. The question about insurance protection, so this law was passed, GINA, G-I-N-A. Anybody heard of that? Sarah, you know? Yeah, what is it?
Isn't it supposed to, it's, if I'm remembering correctly, that it's not supposed to affect whether your insurance covers a condition or not? So like if you have a certain type that is untreatable, your insurance company still has to pay for supportive care even though it's like lethal or something.
You got it. So it's the Genetic Information Non-Discrimination Act. Right, so it basically says that if this kind of information is known about you, you can't be denied health insurance. Okay, so it protects people to some extent, right? You might get denied life insurance. That is, there's still possible ways to discriminate, but at least you can't get denied health insurance. So good. Any other barriers? So why haven't docs ordered this? We talked about the practical barrier, Gary.
Cost, potentially.
Yeah, who's paying for all this, right? Okay, what else?
If the test even exists.
Yeah. Are these tests even available in most hospitals? I can tell you they're not. Anything else?
Patients probably don't know a lot about them either, so.
Yeah, you're so right, Megan.
Informing patients.
Yes, so that, I don't know, tell me what you're thinking about there. That's a really,
I mean, if the doctors already don't know much about them and you come in and they say, oh, I'm going to do this genetic test. I mean, I know I explain even like what I do here in my research here to my parents and they have no idea when I start talking about genes and DNA, and so I mean, trying to explain that to everyone is just, it can be difficult.
great point. So that you have to have then some conversation with the patient about this. And I thought you were actually also going to say about a practical barrier to do that. I mean, how long do you see your doctor for? Real? You were going to say this, Gary?
Yeah, I was just gonna say, like, why do I wanna wait for it, pay for it? You know, what does it matter? This is the medicine for this thing. Like, if they don't know, they're not gonna wanna be inconvenienced by it.
Fair enough. I thought you were also going to talk about sort of the time constraints to do this, right? Most docs' visits now are 6 or 8 minutes, right? Do you have time to spend that, have that discussion? Pardon?
Explain. I mean,
Explain it.
I've been to the doctor once and they didn't even wait for a strep test. They just
Yeah.
gave me an antibiotic.
Yeah.
So with the testing, is it just like a qPCR for like the expression of the gene?
I'll show you. I'll actually show you.
Okay.
Good question. All right, so let's go forward. So we're trying to do this. So we're trying to overcome all the barriers at the University of Chicago. So we created this 1,200 patients project. The initial goal was to enroll 1,200 people. We wanted to have pharmacogenomic testing done, 1,200 patients, and then we would do the test and we'd give the results back to their doctor and then see what happens. So you guys do experiments all the time, right? You're doing them on Drosophila or yeast or mice, right? So this is a human experiment, right? We're asking people, will you be tested? We don't know what results we're going to find. We're going to give them to your doctor and then see what the doctors do. So we actually just wanted to take that first step, right? Because nobody had really done this before, saying, will doctors even look at this information? Will they know what to do with it? Can we help them learn what to do with it? And then will it actually make any difference, right? Will it change what they would prescribe? Would it help patients, get better care? So that was the question.
Are these healthy patients, sick patients, or just a wide range of?
I think I actually have I'll show you. I will show you that. Yes, that's a good question. And so I'm a big believer in that any research you do, you should have a hypothesis, right, that you're going to test. Okay? So that never goes away. So I mean, here I am. I do this for a living. We've got a hypothesis. So I think our hypothesis was straightforward, that if we provide preemptively obtained pharmacogenomic results at the time of prescribing, that we can improve prescribing decisions and patient outcomes, right? Seems pretty straightforward.
For a small set.
For a small set of people, correct.
And a small set of genes, or gene markers.
Correct, and,
Okay.
and a small set of gene markers.
I see.
Correct, that's right. And what do I mean preemptively obtained here? First time I've used that term.
Like You didn't wait for the person to come in with the problem, you just like got a laundry list of things. So.
You got it. So the idea here was, as soon as the patient signs up for the project, they give us a blood sample, they get it tested preemptively for all the markers that we think are important, whether they're on that drug now or not, right? Because is the germline DNA going to change? Right? It's yours for a lifetime. So whether the doc's going to prescribe Simvastatin tomorrow or a year from now or 10 years from now, we've already done the test. And that gets rid of the problem that Dennis, right, you were saying, well, the patient doesn't want to wait for their result. Well, you've already run it. Okay? So that's the idea. So this was our model. We had patients. We had to have patients that were willing to submit DNA samples. We had to have providers who would assimilate the genetics along with everything else that goes into a personalized medicine decision, right? Can't use it in a vacuum. And then we had to have some informatics system that could deliver this at the point of care. And we called that our genomic prescribing system. The GPS, the double meaning is intended, right? We hope that this is helps us get the way to personalized medicine. And then the output is, we hope we're selecting the right drug or the right dose for each patient. Okay. So Megan asked about the participants. So again, you're doing a research study, you're going to identify what animals, type of animals are going to go into your experiment, right? Or what's going to be the makeup of the people being studied. So we had to do that too. So we started with just adults. We didn't want to do this in kids in the first step. Adults who were receiving care at our center, and they had to be on at least one prescription drug, right? I mean, all studies about pharmacogenomics, we want people that are going to potentially, get useful information. And then we had to actually enroll physicians too, right? Because we weren't giving the results back to patients. We were giving the results to their provider to see if they wanted to use this. We never told the doctors what they should do. We never told them they had to do something with the result. They were all free to practice under best clinical care. So we got doctors from all these different specialties. And I will note here that the average number of years in practice for these docs was 20. We had one physician who's been in practice for 46 years. He's actually our most enthusiastic user, right? So you can learn this on the fly. All right. So this is what our test panel looked like. People were asking about. So we ended up creating this on our own. This is a chip that's that size, right? It's filled with hundreds of wells that have individual assays that will assay different polymorphisms in our germline DNA. We end up testing a few hundred markers that we think are important for drug prescribing. Everything we put on the panel has evidence behind it. So we weren't really trying to discover new markers. We were trying to take ones like the ones I've shown you that have been published and seem to be clear for benefiting patients and saying, let's test them and deliver this to docs, because no docs were doing this. Sarah, you brought that up, I think. Do hospitals even have this kind of panel? Our hospital didn't. Most hospitals didn't, so we had to create this.
So is this the microarray? What?
A microarray is typically used when you're talking about gene or expression levels, right? This is what Josiah was asking. So these are actually reactions on your germline DNA to look for polymorphisms, to detect your genotype, okay? CT, TT, CC. That's what you get out of it. You get a genotype. All right, so let's go forward. So I told you we had to have an informatics system to deliver this at the point of care. So we enrolled a bunch of patients. They all gave us blood samples. We tested them preemptively, and then we said, let the experiment begin. Start releasing these results back to docs and see what happens. And so we created this informatics system to deliver results back to doctors. It was a standalone system. The doctors can actually just log into a simple web address, and they would get their patient's results. It was all protected so that only docs in the project could actually see the results. And the patient couldn't get access. They had to get it through their doctor, right? So the doctor logs in. They see a list of patients. All these names are fictionalized, by the way, so I don't break any rules. And the patients that had gray were ones that had signed up for the project but hadn't given us enough blood sample yet. And the ones in blue are the ones that they've signed up and they've given us their sample and we've done the test. And our turnaround time is about a week, actually. Okay? And then we just waited for patients to come back, right? So we used our unit of measure as, well, the patient comes back to see their doctor for a clinic visit. What's the doctor do? Okay. Say the doc clicks on one of those names. This is the screen they get. So this is an actual patient screen with real results. This patient was on 3 different drugs: aspirin, lansoprazole, and hydrochlorothiazide. And we took the most simple initial approach to saying, docs want to know what does the genetics mean in the most basic form. So we use this familiar traffic light symbol. So the green light means the genetics are favorable for that drug, for that patient, that specific patient, right? It means low chance of side effects or a high chance the drug will work. Yellow lights are cautionary, so increased risk of side effects or decreased chance of it working. And then red lights are those really high risk alerts where there could be some, really adverse outcome for the patient based on their genetics. We coded all these as a level of evidence. Level 1 means that the FDA has put this type of information in the drug label. Okay? There's not that many drugs that have really reached level 1. Most of them are level 2, where there's been multiple studies showing this, demonstrating this genetic association with this outcome. And so it's still based on usually thousands of patients being studied. And so we tell them the level of evidence. And then this is the best column. Zoe, you were getting at this, and other people have mentioned it. So, well, if I've got a yellow light, is there actually an alternative I could use, right? So this is the favorite column of the docs. Because we've done preemptive testing, right? We've tested all the drugs up front on the panel. So I can tell you that this yellow light actually is, this is a blood pressure lowering drug, hydrochlorothiazide. I can tell you the yellow light here actually means that the drug probably doesn't work as well at lowering blood pressure. So if you're the doc, you see this result, you're saying, you know what, my patient's blood pressure isn't very well controlled. Maybe I should switch them to another drug. We've put in here 8 other blood pressure lowering drugs that for this patient are genetically favorable. So that's the idea. And we don't just give the traffic light, so any one of those, they could click on the traffic light, they get this, which is a more detailed summary of the information. We thought this was so important, right? You could just give them a genotype, right? It took us 10 minutes to work our way through that Simvastatin graph, right? Imagine a doc trying to do that in the clinic, right? They want to know what's the bottom line. So we actually devised these originally as 30-second summaries. We thought in a busy clinic, 30 seconds or less, they have to be able to read them. And we actually timed it, right? We would time how long they would spend on these pages as they're clicking, and nobody would read more than 30 seconds. So we were really happy. And then we figured out that, you know, as we pre-testing our docs for this, they said 30 seconds is way too long in a busy clinic. We need the 5-second summary, right? And so then we created the box, and it actually tells them exactly what to do. It says, your patient has a genotype that confers a deficient enzyme status, and fatal toxicity due to myelosuppression could occur if the drug is used. Dose reduced to 10% of the normal starting dose. Okay? So it tells them, bottom line, here's what you do for this patient. And remember, for a different patient, their life could be completely different based on their genetics. Okay? I know we have to finish up here, so here's what we've shown so far is that we've approached over 1,200 patients. Almost all patients are enthusiastic about wanting to do this. 87% say yes, they consent. So we've genotyped almost 900 patients now. We've evaluated this now in over 2,000 different clinic visits, so the patient's coming back to their doctor, and then we see what happens. So we didn't even know if docs would log into the system, right? So this is potentially a barrier. 76% of the time they actually log in at the visit, and this number hasn't gone down over time, which we're really happy about. We've delivered all kinds of lights, you know, across the population. Very few red lights, mostly green lights. And these were all on drugs the patients were already taking before the genetics were even available. Why would this be?
Well, if there's like a negative side effect, they might have been on the drug already, and then they were like, ah, it's not working, or I feel sick, so they got switched to whichever one that worked.
You got it. The patients and docs had already figured it out, right? By trial and error, probably. So these were on the drugs they were already taking. So what actually did the docs do? So how often did they click the traffic light to read the 30-second summary? Thankfully, 100% of the time for the red lights, right? Most of the time for the yellow lights, and then less time for the green lights. And we've asked them about this, and they say, it's green light, we're fine. Yeah, we're good with that. We don't have time to read any details, nor do we need to, right? And then we wanted to know, well, how often do they change what they're doing, right? So how often, like you were asking, Zoe and others, would they actually change to another drug? So most of the time for the red lights, they stop the red light drug, okay? And people always ask me, well, why isn't it 100% of the time here? So it turns out that most of these red lights were for drugs in a class called proton pump inhibitors. They treat heartburn. And the red light is that the drug is quickly metabolized in your blood, basically to almost minimal 0 levels. Okay, so the red light is that this drug probably isn't effective for your patient because it's being rapidly metabolized away. And the docs say, well, we saw the red light, we went and asked the patient, and the patient said they were doing fine with their heartburn on this drug, so we kept them on it, which is entirely appropriate, right? So they use the clinical information to talk to the patient. What we do think is interesting is that the yellow light change, how often they stop these yellow lights, seems to be higher than green here. And why would they change green light drugs? People always ask me, I mean, this isn't for genetic reasons, right? The patient comes in, they say they want to stop a medicine, or they can't afford it any longer, or the formulary changes. So we think that green lights is sort of your basal rate of drug change, and that actually is borne out by, there's a bunch of drugs that just don't have pharmacogenomic information. It's just the state of the field, right? Nothing's been discovered. And the basal rate of change of those is also at 8 or 9%. So docs seem to be reacting to this, you know, information. So let's.
Does anybody have class? Okay. So if you have to leave for, you know,
Yeah.
reason, feel free to leave. But
Yeah,
It's been a little bit over, yeah.
Yeah, let's wrap it
But
up.
this is great.
So I've shown you a little bit of the early data, right? But we've been doing this now for 3 years at one institution, and our response rates have been really enthusiastic. I can say that our question of whether docs would even use this, our answer so far is yes. They love using it. They want to keep coming back to the system.
And this is all at the University of
At
Chicago?
the University of Chicago,
Okay.
right? We've done it in about, almost 1,000 patients now. So our patient interest has been robust. Our physician adoption of this information has been high. The alerts that we've been getting seem applicable to patients. I had it on one of the slides there that 90% of our patients have at least one drug where we were able to give a pharmacogenomic signal. So it seems applicable to most people sitting in the room, or most patients at least that were being seen in our clinic. And then these results that we were giving were utilized often. They actually influenced medication choices and informed prescribing. So in your research, right, you always have to ask what's next, right? So what are you going to do? What's the next experiment, right? So what's our future directions? You can imagine that, well, we need to study this in a larger number of patients and physicians. We need to look at this in more than one institution, right? You can always have bias within one place. And what we really haven't shown yet is, did we really change outcomes, right? We've shown maybe we changed what physicians' behaviors will be. They've stopped medicines. But did we actually prevent adverse events? Did we actually increase response rate? So you need a much larger study to really show that. And then people will also ask me, well, what if giving this information to doctors isn't the right place? What if it's a pharmacist? What if you, before you pick that prescription up at Walgreens, maybe the pharmacist would say, hey, do you know you have a red light? So we've thought about other ways to integrate this into the healthcare system to help most people. And I'll end with this. So what about each of us, right? When you think about the global picture of prescribing and all the drugs that are out there and all the genetic markers that we all carry, we've actually done this analysis that it turns out that about 1/3 of us, we have a red light for some medication that we might be prescribed during our lifetime. And 3/4 of us have a yellow light for at least one drug. So I would argue that this kind of information would probably touch almost all of us in the room, and it's something that we really are going to, I think, want to use as standard practice someday in medicine. So I'll stop there. You guys have really been great in answering questions, and we can ask questions or